Today we're going to be looking at Module 10 over protists and fungi. So we'll begin with what is a protist? They are eukaryotes, so they have a nucleus, but they don't really fit into any of the other three kingdoms in domain Eukarya. The other three kingdoms being fungi, plantae, or animalia. So if we're looking at Kingdom Protista, almost a catch-all kingdom, they end up representing a wide range of shapes and functions. They're organisms that are primarily unicellular and eukaryotic, but they're varied. There are over 200,000 different species of protists. And while most are unicellular, there's even exceptions to that rule. There are quite a few that exist together in colonies or as many cells in association, and others are even just multicellular. So a typical eukaryotic cell structure is still what we see for protists. They are in domain Eukarya, so regardless what kingdom or whether or not that kingdom is a catch-all kingdom, they are still cells that have a true nucleus. But they are considered simpler than the other eukaryotic kingdoms. And they're considered the simplest eukaryotic organisms because most are unicellular. Now, traditionally, protists, they've been grouped by how they obtain nutrition. Like, are they autotrophic or heterotrophic? There are animal like protists and fungus like protists that are both heterotrophic. Animal like protists are generally called protozoans. Fungus like protists are mainly heterotrophic, but they're going to be looking at other organisms that they consume that are already dead. They're decomposers, so they're mostly feeding on, again, dead and decaying matter. The plant-like protists are autotrophic, meaning they can convert sunlight into chemical energy or produce their own food via photosynthesis. And these are the ones commonly called algae. Remember that heterotrophic means an organism eats other organisms to convert that energy into chemical energy. And we'll see further classifications based on their locomotion. So protozoans, the animal-like protists, they actually have a mechanism that allows them to move around. Maybe it's cilia or a flagellum. And we don't really see that type of mobility in fungus-like and plant-like protists. There are, of course, exceptions. There's the genus Volvox and Euglena, which are in the plant-like and algae category because they are autotrophic, meaning they can photosynthesize or use sunlight to create chemical energy. And even though their nutrition puts them in those categories, they can move. They are motile. So they don't quite fit locomotive wise. So let's begin with protozoans, the animal like protists. There's three groups of protists in this category: protozoans with pseudopods or false feet, protozoans with cilia, also known as ciliates, and protozoans with flagella. So all of these protozoans are animal-like. We're going to see that in their motility, whether it's through false feet, cilia, or flagella by which they move. So we're going to begin with the ones with pseudopods, and we'll look at the sarcodines. And the sarcodines are a group of protozoans that move and feed by using the false feet called pseudopods. So Sarcodinia is a phylum. So that is an organizational class that consists of members of protozoans that creep across on mud, on the bottom of a pond, over wet plants and rocks. And amoebas are the quintessential example of a sarcodine, using their pseudopods to move about in their environment. They are representative of protozoans with pseudopodia. So a pseudopod is just a false foot. It's a temporary bulge of the cell membrane, and amoebas can push these out in order to have their cell membrane and therefore themselves move throughout its environment. They tend to inhabit watery environments. They don't want a lot of current because while they can move, they're not very strong swimmers. So more stationary water like ponds and lakes, slow-moving rivers, those would be areas where you may find an amoeba. They don't swim, they are just motile. They can cling, they can move about in limited space, And if you've ever put your hand on a slimy rock, then you've probably touched an amoeba. They're using their false feet for locomotion, but also ingestion. They can take those pseudopodia and extend them around their prey and take it in or engulf it. This is a process called phagocytosis. And once they have their prey engulfed within their plasma membrane, they enclose it in a food vacuole in order to break it down. Just like plant cells have water vacuoles, the amoebas are going to have contractile food vacuoles. They may also have those that help regulate their internal pressure compared to their external pressure. In that way, a contractile vacuole may help regulate cell structure by collecting extra water from the cytoplasm and then expelling it from the cell. And that's going to make many amoebas hypertonic compared to their environment, meaning inside them they're going to have more solute dissolved in a solution or solvent. So they're going to have a higher concentration of substances within their solvents than what we see in their common environment. So they have to have contractile vacuoles to handle the extra water pressure, or else they would explode. Next, let's move on and look at the flagellates, like the zooflagellates. Still protozoans, so still animal-like protists. And a zooflagellate is an animal, like protist, that uses flagella. So pretty basic there. Zooflagellate, think zoo, animals. Flagellate, think flagellum, so it's an animal-like protist that uses a tail to move about in its environment. Flagella are generally long, thin, and whip-like. And we're going to see that a lot of these zooflagellates reproduce asexually by binary fission, meaning they essentially split by copying themselves and produce copies of themselves. And most species of zooflagellates live in lakes and streams, again, more stationary water. While they are motile, they're not great swimmers, and they're going to absorb food through their cell membranes, also via phagocytosis, just like the amoebas. And a couple of different examples of zooflagellates are parasitic. Now, speaking of parasitism, there's different types of symbiosis we see when we're looking at protists. Some of those symbiotic relationships are parasitism, wherein the host is at the detriment to the benefit of the protist. But a base symbiosis is just a close relationship between two organisms where at least one of the organisms benefits. So technically, a predator-prey or parasitic host relationship is a type of symbiosis. So speaking of one type of parasitic zooflagellate, we have here Trypanosoma. Trypanosoma is a zooflagellate, an animal-like protist with a flagellum, and this one tends to be passed to humans by insects. Specifically, the African tsetse fly can carry Trypanosoma protozoa inside of it, and then when they land on and bite humans, they can pass the Trypanosoma protozoa to the human. And that's how humans can become infected if they're bitten by the insect carrying it. They can also become infected if they eat infected meat from an animal that was bitten and had the Trypanosoma in it. So parasitism definitely classifies as a symbiotic relationship. But of course, only one organism is benefiting, and that is generally the parasite. And these protozoans that we've been talking about can also live in mutualistic relationships. One example is protists that are zooflagellates that live in the gut of termites, allowing termites to break down cellulose, and that is a nutrient for the termite, so it helps them survive. There's also Trichonympha, and that is the name of the protists that are zooflagellates that live in certain termites and feed on that cellulose. So Trichonympha is the protist, but protozoans living in the gut of termites is an example of a mutualistic relationship. So without that protozoan, the termite would be unable to digest the wood they eat and they would die of starvation. So mutualism was a type of symbiosis in which both partners benefit from living together, though different, of course, than parasitism. We can now look at protozoans that have cilia, or tiny hairs that they move in a wave-like pattern so that they can move about in their environment. And a ciliate, still animal-like, It's a protist with cilia for motility, and animal-like protists called protozoans that are within the phylum Ciliophora are the ones that use cilia. Ciliates are also some of the larger protozoans. They can get upwards to lengths of 3 millimeters, and they vary in shape. They live mostly in the fresh water of stagnant lakes and ponds, just like all the other protozoans we've been looking at. A model ciliate is the Paramecium, and some of the things that are interesting about them compared to other parameciums or even other model ciliates are that they have very obvious structures such as a macro and micro nucleus. The large nucleus that controls the paramecium's metabolism is the macronucleus, and a lot of energy is required for the rapid movement of their cilia, so therefore they need this macronucleus to regulate their metabolism. And then we have the micronucleus that controls reproduction. That way, the macronucleus can spend all the time controlling metabolism and making sure the cilia move precisely in a wave-like manner that promotes motility. And the micronucleus can be concerned not with that metabolism, but instead reproduction. Ciliates are also going to have a contractile vacuole, like we saw for some of the other protozoans. They have food vacuoles to hold food while it's being digested. The way that a paramecium as a model ciliate would get food is to use an oral groove that's lined with even more cilia to sweep or suck food into a groove. This groove is called a gullet, and it ends in an oral pore, and that pore will fill with food, and that will be when the gullet pinches off to become a complete food vacuole. Any undigested food would be expelled through an anal pore. So for some definition here, cilia are the hair-like projections on the outside of cells that move in a wave-like manner. So for all the protozoans we've been looking at, we've been focusing on their motility. And their motility makes them animal-like. The fact they can move about in their environment, whether it's with pseudopodia, flagellum, or cilia, gives them that animal-like nature. Let's also look at how a paramecium would reproduce. Conjugation is common, and that would be a process wherein two paramecia attach to one another at their openings or the oral grooves, and we would see micronuclei reorganization of the genetic material and some DNA exchange. And it's that DNA exchange that makes it conjugation. A mutual exchange of DNA would be seen, and we see that in ciliates, not bacteria. So it's slightly modified from bacterial conjugation. We still have an exchange of DNA, just in this case with the paramecium, it's mutual exchange. Then after the conjugation, the two paramecia would separate and then immediately reproduce asexually via binary fission. So you'd have resultant new paramecia that, because of the blending of DNA after the mutual exchange, will be slightly different than their parents. Other ciliates we can talk about besides paramecium are stentors and the Balantidium coli. Now, the genus Stentor contains organisms that can grow as large as 1/10 of an inch, which is large for ciliates. And they're going to be also common in freshwater habitats. They kind of look like a trumpet. They're wider at one end than the other, and they have a much rounder gullet, and they would have cilia surrounding that gullet. And they can eat all kinds of things, including paramecia, which is another ciliate, and bacteria. The Balantidium coli is pathogenic. It can cause disease. It's a common parasite of pigs, rats, guinea pigs, and as a ciliate, it can also form cysts that can survive in fecal matter and then infect humans if we happen to eat or drink contaminated food. An infection with B. coli is not as common in the United States as infection with E. coli, but B. coli is a common infection of humans in tropical regions of the world. Next, we're going to be looking at just a few examples of non-motile protozoans. So protozoans are primarily animal-like, so these kind of don't fit the norm. And the reason they're non-motile is because they're all parasitic. They're not motile in their environment because their environment is a host cell, and they are classified in the phylum Sporozoa. There's more than 30,000 species in this phylum, and they all form spores, hence the name sporozoan, and at some point in their life, these spores are produced. and they generally feed on the body fluids of the host in which they are in. So parasitic protists are sporozoans. They feed on cells and body fluids of their host. A spore would be a tiny cell that is able to grow into a new organism, and they can be produced by all sporozoans, at least in some point of their life cycle. A lot of these can hide from the host immune system by acting like a cyst. Their spores can essentially hide or create structures around them that are protective. And these spores can also reproduce asexually by copying themselves, wherein the nucleus divides several times in a single cell. The cytoplasm concentrates around each nucleus. We get hardened shells of cytoplasm, and we can put or group together several spores. And the cell may then disintegrate, allowing the spore to be released so they can grow into a new sporozoan. So oftentimes they'll use your own cell's machinery to hide. There are some exceptions to spore formation when we're looking at protists that we consider protozoans, and that would be those in the genus Plasmodium. They are sporozoans, but they do not produce what you would consider spores. They're still parasites, so they fit very well within sporozoan. They're spread by mosquitoes, and their life cycle is as follows. They produce spore-like structures in the gut of a female mosquito. The spores then travel to the salivary glands. The spores then enter the bloodstream and travel to the liver of a human when the mosquito bites them. Once in the liver, the Plasmodium spores become new Plasmodium, and they reproduce asexually. And some of these offspring stay in the liver and cause future episodes of malaria. Now, some Plasmodium venture back into the bloodstream, and the Plasmodium invade red blood cells in order to reproduce asexually until the red blood cell bursts. If an uninfected mosquito bites an infected human, Plasmodium enter the mosquito through the blood it is taking, and that's going to allow for further spread of the infection. So Plasmodium, that genus, is in the phylum Sporozoans. They cause malaria, and while what they pass from host organism to host organism is called a spore, it's not a spore in the same way that we would consider the spores of the other pathogenic sporozoans a spore, where it's a tiny cell that is able to grow into a new organism. We actually see it pass from host cell to host cell and not really grow into a new organism, but essentially reproduce asexually, just copy itself, and have the spores be in abundance. With the life cycle of Plasmodium, you may have noticed a back and forth type of reproduction, where these spores not only were undergoing asexual reproduction, but eventually they would create a multicellular diploid organism as well. This life cycle is called alternation of generations. It also is one of the reasons that we would consider Plasmodium an exception. Not only do they create spore-like structures, but that's not all they do. So it's the exception to the spore formation rule, because we're going to use those spores to create a multicellular diploid organism. Alternation of generations is also very common in plants, so you'll learn more about that later. We can also look at Toxoplasma gondii, and its spores are generally sexually produced in the intestines of cats, and not usually harmful to the cats, but they do exit the animal with its feces and are not destroyed. It can also travel via houseflies, cockroaches, insects, and direct contact with cat feces. And once T. gondii infects a human, they reproduce asexually, causing the disease toxoplasmosis. So again, we're seeing kind of an exception to the rule of just spores. We're using the spores here in both asexual and sexual production. And so this is another alternation of generation. Toxoplasmosis as a disease causes severe birth defects. And that's why doctors tell pregnant women that they should never empty litter boxes or otherwise clean up after cats. All right, next we're going to be moving away from the protozoan, or the animal-like protists, and looking at fungus-like protists. Fungus-like protists are heterotrophic, generally live in damp, nutrient-rich environments, and unlike true fungi, these are going to have centrioles, and they're going to lack chitin, which is the main component of the fungi cell wall. So, like fungi, fungus-like protists are heterotroph, have cell walls, and use spores to reproduce. But unlike fungus, they are also able to move. Regular fungi do not move. We're also going to see that the cell wall of fungus-like protists does not contain chitin, and chitin is in fungi cell walls. There's two types of fungus-like protists, slime molds and water molds. Let's look at slime molds first. Slime molds are found where there is moist, decaying organic matter. A lot of slime molds are decomposers. You might find them on a compost pile or on dead trees in the forest. They're playing a key role in recycling nutrients. There's two broad groups of slime molds, both cellular and acellular slime molds. Cellular slime molds look like soil amoebas. They spend most of their life as free-living individual cells. They're going to be looking for a food supply, and they'll move on when that food supply is exhausted. And cells will generally send out chemical signals to other cells nearby to aggregate or figure out if they should move their kind of large slug-like body to that area. They can form colonies, and colonies could contain thousands of individual cells, but it does begin as the colony forms to function like a single organism. These colonies produce fruiting bodies, which are reproductive structures that produce spores. And when the fruiting bodies release spores, they'll be carried away and become new amoeboid cells if there is food readily available. So here are some examples of fruiting bodies. In a true fungus, the mushroom is the fruiting body, a reproductive structure that produces spores. These are slightly modified because they're in fungus-like protists, not true fungi. Acellular slime molds have an eating stage wherein they have individual amoeba-like cells, but as they aggregate, they fuse together so that there is no longer cell membranes separating these cells into separate individual cells. They become one large cell with many nuclei. This structure of the removal of cell membranes and the creation of a multinucleated cell is a plasmodium, not to be confused with the type of protozoan that causes malaria. But a plasmodium, once it's formed, can move about in search of food by sending out pseudopodia. So the plasmodium will stop moving when food or water become exhausted and produce fruiting bodies, and the spores produced from the reproductive fruiting bodies can germinate into flagellate cells, which can then fuse to start the cycle again. Here are examples of slime molds, and I will post this PowerPoint on Teams. If you open it up in Microsoft PowerPoint, you'll actually see that these images move, and they are going to be kind of pulsating amoeboid-like movement. Water molds and mildews are something we'll look at next, and they decompose dead plants and animals in freshwater environments. So they're also going to be responsible for recycling nutrients in a habitat. Some water molds are parasitic and grow on the gills of fish. Others can grow on other substances like different types of plants. The plant destroyer is one example of some water molds and mildews, but not all water molds and mildews are parasitic or are going to destroy other types of life. Water molds can exist as individual cells or fuse to form thin filaments called hyphae. Hyphae are similar to what we see in true fungi, but here they do not have walls or cell walls between the cells, so they are more like plasmodia. Even though they're multinucleate, they're not going to have the same cell wall, cell structure, or have chitin like true fungi do. Water molds do have both asexual and sexual forms of reproduction, and so we see kind of that dichotomy in reproduction like we saw with the alternation of generations. Water molds that grow on land are called mildews, and a lot of them are plant parasites. Even though not all of them are, a lot of mildews can cause plant blights, such as the Irish potato famine of 1845, which was caused by Phytophthora infestans, which is a water mold that attacks potatoes. All right, next we're going to be looking at plant-like protists like the euglena and the algae. Euglena and algae are plant-like protists that contain chloroplasts. That's putting them in that category, plant-like protists. The chloroplasts allow them to be autotrophic. They can perform photosynthesis. They can make their own food. They're found near bodies of water and any area that you might find algae. And algae can be found in both marine and freshwater environments. A lot of times, this algae gets called plankton or phytoplankton, and those are all just tiny organisms that float in the water. You can categorize the plankton into two groups, zooplankton or phytoplankton. Now, the reason I said commonly called phytoplankton is because we're now looking at plant-like protists, and phytoplankton is floating photosynthetic organisms like the plant-like protists, euglena, and algae. Zooplankton also can make up the broader group of plankton, but those are going to be the protozoans, or the animal-like protists. Photosynthetic phytoplankton, or algae, are autotrophic and produce most of the oxygen on Earth. It's actually not produced by terrestrial plants. Instead, it's being produced by these photosynthetic autotrophs, or phytoplankton. Algae are definitely a major source for food for many aquatic organisms, and certain types of marine algae are even eaten as vegetables or used as food additives in certain parts of the world. Carrageenan is a substance extracted from Irish moss, and it's actually used to thicken ice cream. That's a type of phytoplankton that's used as a food stabilizer in not only ice cream but sauces and dressings. It also can be used as a natural kind of fertilizer. If you like sushi, different types of algae become the nori that wrap a lot of sushi. And iodine, nitrogen, vitamins, and minerals can even be made from processed algae. So let's look at some more protist vocab. Carrageenan, which is extracted from red algae, is used in products such as ice creams and hair conditioners. It is a product stabilizer. More on phytoplankton. A lot of the algae that we call phytoplankton exist in individual simple forms, like cells by themselves, but a lot of them can form colonies, and they're held together with slime. A few species form complex colonies, but whether it's individual or in colonies, algae can reproduce both sexually and asexually, so it does demonstrate the alternation of generation. There are some differences again between the cell wall of phytoplankton and that of true plants. Their structure and the type of photosynthetic pigments they possess does differ as well. Euglena are one of the examples of a plant-like motile protist, and they're found in freshwater lakes all over the world. They do contain chloroplasts but no cell wall, so they can perform photosynthesis. They have one or two flagella, and they're also saprophytic, so they absorb their food. So they display characteristics of both animal and plant-like protists, so it was the presence of chloroplasts that got them put in the plant-like protist category. But scientists did have a difficult time agreeing on which phylum they should be placed in. Because like the sarcodines, like the ciliates, they have contractile vacuoles. They're also mobile, like many protozoans. They have a nucleus that contains DNA that controls metabolism and reproduction. And while they have no cell wall, they do have a pellicle, or a flexible coating outside the plasma membrane that helps the euglena maintain its shape. And that's something extra we don't see in the protozoans. So that made it, with the addition of the chloroplast, more plant-like. They do also generally have a red spot. We know this is light sensitive. It's kind of like a primitive eye spot. Euglenas are going to use their eye spot to move toward regions of bright light, and that helps them increase their photosynthetic output. And we also know that euglena produce only asexually, so they're kind of an exception to the rule of alternation of generations. They produce by binary fission only. We can also look at other algae called dinoflagellates. A lot of them are photosynthetic and heterotrophic. They're usually unicellular protists with flagella. They have cell walls made up of cellulose, like plants. They're abundant members of the phytoplankton or photosynthetic protist group, and they can be found in fresh and saltwater environments. The term dinoflagellate comes from the Greek word dinos, meaning whirling, and they use that flagellum that whirls them as something that makes them motile. The name dinoflagellate comes from the fact that most species have two flagellum. So not only are we saying that dinos gives them an idea of how they're moving with a whirling motion of their flagellum, the fact that they're called dinoflagellates tells you they have two tails. Dinoflagellates reproduce mainly by binary fission, just like if we go back and look at euglena, and euglena only produce asexually. Dinoflagellates also produce by asexual binary fission. We do see that sometimes, thanks to things like nutrient runoff in the Gulf of Mexico, we have algal blooms. Essentially, many dinoflagellates being created. A population boom in this respect can be damaging, however, because certain species of dinoflagellates produce toxins, like that of Gymnodinium breve, and those toxins produced when there is a population boom, thanks to nutrient runoff, can create an abundance of these toxins in water and kill aquatic life, including turtles, manatees, dolphins, and it can be even damaging for many fish and also humans who happen to be on the beaches where these red tides are located. They're called red tides because of the pigment or hue that the toxins with the population boom tend to take on, which is anywhere from a red to an orangish hue. Now, an algal bloom doesn't have to be red. An algal bloom could just be a population boom of algae, green algae, brown algae, but red tides are most dangerous because of the toxin produced thanks to the rapid growth of a population of algae. The red tide, again, is an algal bloom that occurs only in marine environments. They're deadly to marine creatures because of toxins emitted into the water in mass by certain types of dangerous dinoflagellates. Normally, the toxins would not reach deadly proportions, but during a population boom called an algal bloom, these toxins can reach the deadly proportions that can kill clams, oysters, and mollusks, but even bigger vertebrates like dolphins and manatees. So if you wanted to test yourself, you could explain, why are red tides dangerous? We know that they're an algal bloom, that they're caused by the overproduction of phytoplankton like dinoflagellates, thanks to nutrient runoff. They can be very dangerous to vertebrates, and that's because the toxins that the algae produces may lead to serious illness or death to organisms that consume the algae. And something a little bit more fun than the deadly nature of some dinoflagellates is those that have luminescence, or bioluminescence. Many dinoflagellates have the ability to produce light called luminescence, and that's when they're disturbed by either people walking in the water or by oars or crashing waves. They give off this eerie blue glow. Eutrophication is the term I've been using for one of the reasons you may see algal blooms, and that is the buildup over time of nutrients in freshwater lakes and ponds that eventually can lead to an increase in the growth of algae. Sometimes this buildup of nutrients in these ponds and lakes can run off into marine environments like the ocean, and that's where it ends up producing red tides. Some of the main triggers for eutrophication are fertilizers spread on fields that can make their way to nearby lakes and ponds, and poorly designed or aging septic tanks that leak their contents into the soil. All of these are producing excess nutrients that oftentimes algal blooms do not need, and it leads to the population boom or an increase in population, and in the case of red tides, an increase in toxins. Next, we're going to look at golden algae. So we're looking at diatoms that belong to the phylum Chrysophyta. There's many different types of diatoms. They're one of the greatest producers of oxygen in creation and one of the most abundant organisms on Earth. They're unique because they have silicon dioxide in their cell walls, which makes them glass-like in appearance. And silicon dioxide as the principal component of glass is going to be very hard, but provide an excellent protection. They're kind of like the stained glass of the Protus world. The cell wall is actually so hard that it remains kind of like a skeleton, way longer than the diatom living within will last. So large amounts of that leftover diatom cell wall, the silica dioxide, can actually crumble and form an abrasive substance we call diatomaceous earth. And that's actually going to be used in grinding and polishing. For example, it's used in toothpaste to polish your teeth. And then diatoms are also able to convert excess food into oil instead of carbohydrates. So there's interest in researching them in order to see if we can use them to make biofuel. Here are just some examples of our glass-like diatoms. They come in many shapes and structures. And again, they are a type of plant-like protus, or an algae. Let's look at some more notes. Dinoflagellates and diatoms are two types of algae that bloom in red tides. So they are the ones that can produce toxins. They do not all produce toxins all the time, but again, with an algal bloom, thanks to eutrophication, it can be deadly to vertebrates and other animals. Next, we're going to be looking at other members of Chrysophyta. Again, that was the golden or yellow-green algae, and a lot of them are going to have chloroplasts, and those chloroplasts will not be green, they'll contain bright yellow pigments, hence the name Chrysophyta or golden plant. We see that alternation of generations or asexual and sexual reproduction in the other members of, or plenty of members for those of Chrysophyta. Most are unicellular, but some form colonies, such as diatoms. But some colonies of Chrysophyta that we're talking about are mainly those that we would consider kind of sessile kelp colonies or multicellular algae. And they would use holdfasts in order to hold themselves or anchor themselves to a location. And the use of holdfasts by colonies of Chrysophyta creates colonies that don't move about, so they're considered sessile. And so a sessile colony is a colony that uses holdfast to anchor itself to an object. Next, we're going to look at green algae. There are over 7,000 species in Chlorophyta, and a lot of them are going to be considered not only algae, but algae that contain chlorophyll with a green pigment, just like the chloroplasts that we find in plants. A lot of them will also have carotenoids, which are orange to yellow pigments, and they can mask some of the green color that you see in green algae. And they also have cell walls that contain cellulose, just like plants, and it could be unicellular, colonial, or multicellular. Here is one of the largest green algae, and that would be seaweeds. Seaweeds oftentimes float about. They do not all use holdfast, whereas kelps are generally going to use holdfast to become a sessile colony. One example of green algae is Spirogyra. It's pretty interesting green algae genera, and that's because not only do they have chloroplasts, they have spiral chloroplasts. And they're going to use their spiral chloroplasts in order to photosynthesize or take light energy and convert it into chemical energy. They're also very slender or chain-like, and we see the spiral chloroplasts wrap around their structure. Volvox is not long and cylindrical. Instead, Volvox is going to be very spherical. It's usually single-celled, and it has organisms within it that form colonies. It reproduces primarily asexually, but does produce daughter colonies, and once they're formed, they generally stay with the parent colony. So while the asexual production is what occurs, essentially we build a colony by the conglomeration of those daughter cells produced by the parent. Under certain conditions, members of Volvox colonies can reproduce sexually, but asexually is much more common. So when you're looking at Volvox, these smaller balls are the daughter colonies, and the larger ball is the parent colony. So here we can see multiple examples of daughter colonies within parent colonies for Volvox. Ulva is sea lettuce, and it looks exactly like that. It's multicellular green algae. It's considered a seaweed because it not only grows large, it also will use holdfast. It has a leafy appearance, and that's why it's garnered the moniker sea lettuce. It is green algae, so it contains chlorophyll in its chloroplast, and it is a plant-like protist, so it does have cellulose in its cell walls. Here is sea lettuce or ulva, so you can see it does fit its moniker. Red algae is in Rhodophyta. That red color comes from photosynthetic pigments called phycobilins, as opposed to chlorophyll and carotenoids. And members of this phylum are generally multicellular and tend to live in marine environments, much closer to the tropics, so in warm water. The red algae can live in the deepest waters of the ocean. We commonly see it associated with coral reefs. And of all the algae, red algae can live in the deepest parts of the ocean, sometimes even next to thermal vents. Brown algae is in Phaeophyta as a phylum. It's actually yellow-brown in color, and that's due to another accessory pigment, which is fucoxanthin, and that's going to be in addition to chlorophyll. So we don't see the absence of chlorophyll here, just the addition of fucoxanthin. And this is about a 1,500 species phylum. They tend to be multicellular, and they like the colder ocean water habitats as opposed to the warmer water habitats that red algae prefers. Just like when we talked about green algae, members of this phylum may look like seaweed, but they're also used in many food products like ice cream, pudding, salad dressing, or jelly beans. They would all use algin, which is a component of brown algae that's used as a thickening agent, kind of similar to carrageenan when we were looking at different types of algae earlier. It's another stabilizing product. Algin is also used in cough syrup, toothpaste, cosmetics, paper, and floor polish. Keep in mind something that is a seaweed is not going to be attached, as well as something that is a kelp. They use holdfast or rudimentary aquatic roots to allow them to anchor themselves to rocks at the bottom of the ocean. Some kelps can grow as long as 100 meters, and kelp is harvested in many parts of the world for food. And a lot of places will actually grow kelp forests, and they're great ecosystems for many marine creatures. And if they are messed with, they are reducing ecosystem habitat variety, and that can damage the ecosystems for those marine creatures. So here is an example of a kelp forest. You can see there is aquatic life that lives within the kelp that is using holdfast to stay in place. We can also look at the genus Fucus, and it's commonly not called fucus, it's called rockweed or bladderwhack, because not only do these algae have a thick, leathery feel, they live in shallow waters, and they have these bladders or spaces that fill with air in order to allow the organism to float on top of the water. And that is it for part A related to Protists. So make sure you're reading this chapter and staying up with the topic. We'll look at part B next Tuesday in class. Prepare to either talk more about Protists and later fungi in class on Friday or do an experiment.